Hello, I'm back. Welcome to another episode of Fiveable AP Biology with me, Jessica Nadzam. I even got my Fiveable swag on today. I came prepared and ready to go. So I can already see we've got about 12 people here. I recognize many names. Sierra, so glad you're here. Nicole, yay, so glad you guys are here. I'm sure a few more will trickle in. You can't hear me? Can anyone else not hear me? Hi, Kenny. Okay, Olivia, I'm a little worried it might just be you, because the others can hear me. Yeah. Olivia, I would try restarting your browser. I'm so sorry. I would give you like captions if I could. I'll have a PowerPoint in a minute, but it doesn't have like a script, so I would try restart. She can't hear me. Why am I saying this? So Olivia, I'm so sorry. Try restarting your browser. All the things that I was saying, thinking I'm giving such good advice, and she cannot hear because yeah, that sounds like something I would do. So woo. Okay. So like I said, my name is Jessica Nadzam. I am a 5th year AP biology teacher. Obviously, this year is a little different than previous years. As some of y'all may have heard, maybe, AP biology, yay, I'm so glad it works, Olivia. AP biology has changed in that only units 1 through 6 will be tested this year as a result of COVID-19 testing. So we're switching gears, which is why my slide over here, that one, says we're talking about community ecology and ecology math today. We ain't, because ecology is no longer on the AP exam. They have removed it for the sake of so many people being out of school. It's not fair for them to try to learn it over the next 6 weeks while they're not in school. And so the theory is that everyone should already have units 1 through 6. I know sometimes teachers teach a little bit backwards. Like, for example, I start with ecology, but luckily we had just finished unit 6. So needless to say, that's why the title of this slide doesn't match what we're doing today. So let's talk about what we're actually doing today. Oh, don't forget, with all this free time that a lot of us have now, Fiveable has tons and tons and tons and tons of streams in the evenings. And we've recently expanded. We're doing a ton of Q&As and other test support in the mornings. Again, our services are free, so I strongly recommend using those like you are now because you're so good. But if you're looking for really targeted support, you can go to the Q&A sessions and be like, hi, help me. And they'll be like, okay, I got you. Or if you just want to do review, kind of like we are here, this is a great option as well. So great job to y'all. There's the schedule. Y'all can read. Don't forget, we're on the internet. We're all millennials or younger here, right? Maybe there's another teacher on here who's older, but I think most of us are familiar with these things. So if I was you, I would go into my Twitter or my Facebook or my Instagram or my YouTube or whatever other app you got and look up @ThinkFiveable and follow it, because we put out tons of announcements and things, and we're closely following what happens with AP. So if you want to get like really quick announcements on what's happening with testing in terms of dates and content and things like that, follow us. So also, we just like to think we're cool, and we're millennials, so like we need likes like everyone else. So on from that blurb, here's what we're actually going to talk about today, since we're not talking about ecology. My goal today is that you are able to complete certain FRQ question types, because in case you did not hear this either, the AP exam is now entirely FRQ. There are no more multiple choice questions. My understanding is that this is in order to cut down on cheating, because it's harder to cheat on a free response test. It's also only 45 minutes, which means there won't be very many questions. Again, short time frame, harder to forge your work, perhaps. So that's my educated guess on why they've made it so short. And that's what Trevor Packer said. Yeah, Trevor Packer is like the Brad Pitt of AP. But So that's why they're doing it. Also, only 45 minutes because people will be testing from home. And frankly, testing from home is stressful. I currently am in like an online grad school program, and it is stressful doing all that stuff at home. Like, it's not as nice as people think it is. So that's why they're making it shorter. As a result, it's all FRQs. I am an AP reader. I have gone and I have graded AP tests before. So I like to think I know what I'm doing. I like to think that. But I have a very good understanding of how FRQs work. And for that reason, I really want to focus on the type of FRQs you can expect from the first 2 units, Unit 1 and Unit 2. If you remember, Unit 1 was Chemistry of Life, which was like biomolecules, water, polarity, all that stuff. Unit 2 was Structure and Function. So that's where you got into like membranes and hypertonic and osmoregulation and all these other buzzwords. You're like, oh no, not that again. That's not what I got into this for. But things you got to know, because they love to throw those kind of questions on FRQs. And now the test is only FRQ. So that's what we're going to do. Japesh, is it also true it's open book? I honestly don't know. I've heard that a lot. And I mean, to be perfectly honest, like, obviously you should never cheat. But the reality is I don't know if AP is going to have a way to police it, which makes me think they'll just make the questions really hard so that even if you have it open book, there's like, it's not going to really help, if that makes sense. You just have to know your content. Like, you won't have time to go flipping through your book trying to find the information. Because if you remember with AP is not a memorization game. AP is application, which is what we're going to look at in these FRQs today. So it's mostly analysis and conclusion based, I think. Yes. So AP is all application analysis. It's not going to ask you, what are the organelles of the cell? Like, that's memorization. You can look that up in a book. So it's not really like the kind of thing an open book would help you, because you have to know your stuff well enough that you already have it memorized so that you can apply it to answer the question. And we'll see that today. So today we're going to kind of review that content that you should have memorized. We're going to talk about properties of water, cell membranes, membrane transport, tonicity. That's like hypertonic, isotonic, hypotonic. And then we're going to talk about a couple of common lab questions that I've seen. Some of them are older just because only older lab questions are released. So I acknowledge the formatting isn't great on these, and you may not have seen them before just because they're old. But I feel like it's valuable for you to see because it's the type of question they're going to give. It's FRQ. And who knows what AP is going to pull out of their hat. Plus, these are very applicable to you guys because some of you should have done labs over these before. So we're going to talk about them. So the first one is going to be, or what I want you to think of when you think of those lab-based questions. Did anyone here do the experiment where your teacher gave you that like plastic tube and you had to like rub it to open it and you had to put like starch in it, but the starch always got everywhere? It was called dialysis tubing. Did anyone do that lab in class? Yep. Whoop, whoop. Kenny, where are you? I know you did it because you're in my class. Kenny, you did it. You're in my class. The dialysis tubing experiment, it's the one where your teacher would give you the little tube. You'd have to like roll it open and then tie it, yes, virtually, and put like some starch in, and it would make a total mess, but then you would put it in some water and you'd watch as things move in and out and That's a really common experiment that we do for labs because it's very hands-on and you see the results relatively quickly. It's also a question they like to put on the AP test. So when we're going through this review, you may be thinking, this is kind of like elementary. I learned this in like August and I know it. I'm not saying you don't know it. I'm saying this is the stuff you need to know to answer these types of questions. Yes, you need to know it. You need to memorize it. You also need to know how to apply it. That's what we're doing. So let's talk about our first kind of unit content area thing that we need to review. We're not doing any specific like units here. If you're looking like, oh, I have no idea how structure and function works, I need a review on that entire unit, you need to either go back to our old videos or better yet, you need to look at one of our cram passes. We have an awesome biology teacher who is currently teaching the cram pass classes, and you get extra content, extra videos and stuff like that. So if you need more help in a specific unit, you should definitely look into signing up for those. I'm sure you've already gotten emails about those. So we're just going to kind of pick and take little content-y areas out of each unit that apply to this FRQ. The first one we're going to talk about is properties of water. So who is this national treasure that we have on the left side of my PowerPoint? Who is this person? Bill Nye, the science guy. Bill. And what is Bill holding in his hand? Please don't say Mickey Mouse. A water molecule, very good. Okay, what are the two white dots? H, hydrogen, good. What's the red dot? Oxygen. Okay, awesome. So we know water. We have water. Water is the key to life. This should all be reviewed. Main things I need you to get out of this part, though, is that water has certain properties. Those are those things up there in the corner. Adhesion, cohesion, surface tension, heat capacity, freezing properties. There's a couple of others. I can't remember them off the top of my head, but those are the main ones you need to know. Which one is the one that lets like lizards and stuff run across the surface of water? Surface tension, good. Okay. Which one is the one that makes water, solid water, less dense than liquid water? Freezing properties, good. Okay. Which one is the one where water sticks to other water? Cohesion, good. Okay, awesome. Now, all of these properties are totally essential to life. If you don't have them, we would not be here. Like, it wouldn't work. And those properties are enabled by hydrogen bonding. So that's the idea that, like, let me see if I can get it to do it. It may do it, it may not do it. Okay, so like, you know, the white dots are the hydrogen, the red dots are the oxygen. So you can have hydrogen bonding, which is when one hydrogen bonds to a separate oxygen with a very weak attraction. And that is a result of something called what? I'll give you a hint. It has to do with the bear on the bottom of the screen. What do we call it? Why are we able to do hydrogen bonding? What is that property of water that makes it able to do hydrogen bonding? Look at the bear. What kind of bear is it? Polarity, a polar bear. I love to throw polar bears on here anytime I talk about hydrogen bonding because it can sometimes be a little bit like not thrilling. Like my husband walked in while I was making this slideshow and he was like, ugh. And I was like, it's science, respect science. And he was like, so this is why you keep throwing images of polar bears on things. Cool, I get it. And I was like, yes. So speaking of things that are maybe a little bit more dull, I'm not a chemistry person. So like this is not really what people sign up for with AP biology, but it's essential because the reality is if you don't have this, you don't have AP biology because there wouldn't be anything alive. So again, we're looking at polarity here. That's why we got this cute little polar bear to make us happy. Oh, he's so cute. Okay. I am not concerned with you understanding those little like S cursive-y symbols. You don't need to know that. I am concerned with you understanding the positive and the negative. So oxygen is going to be very, very negative in comparison to hydrogen, which is going to be more positive. Why is oxygen more negative? Electronegativity. Very good, Shreya, Japesh. Okay. Oxygen is very electronegative. That means it takes all of the electrons off of another atom, if it can, and it hoards them all for themselves. It never learned how to share. Now, hydrogen only has one electron. So when you take away that electron and the oxygen hoards it, that makes hydrogen very, very positive. As a result, you can take these separate hydrogens and oxygens, and they form these little dotted lines. Those are weak attractions, also called hydrogen bonds. Because they can bond together in this way, you get all of these properties: adhesion, cohesion, surface tension, heat capacity, freezing properties. Basically the reason we can like survive and stuff. I like surviving. So it's pretty cool. Does anyone have any questions about properties of water? I know I'm moving fast, but we still got a couple other slides we need to look at. Does surface tension arise from cohesion? Yeah, because water's binding to water then. I would say I wouldn't put on a test like surface tension is caused by cohesion because technically I guess you could say that's right. I would just list them as separate properties though. Just from what I know from grading the AP test, they're going to be more concerned with you listing those out. This is not the kind of area where they're going to want you to necessarily tie that together. You can, but I'm a little worried that they'd be so picky about the wording that if you mess up one thing, they would count it off. So I would just list them as separate things, and that should be fine. Does that help? Okay, cool. All right. I'm going to go ahead and go on to membrane structure. So there is a ton on this slide because I love to give people headaches. But I'm also trying to move through this pretty quickly, and this should be review. That's why it says review up there. What's this big thing in the left? Kind of looks like a balloon with 2 legs, or a stick man with no arms. A phospholipid, very good. Okay, phospholipids have 2 parts. They have a head and they have 2 tails. Which end is polar? The head is polar. The tail is non-polar. Very good. So let's be really clear here. Polar and non-polar don't mix. That's oil and water. Water is a polar molecule. Okay. Hydrophobic. Very good. That's another word for non-polar. But water is polar. Polar things are able to do all those, at least water, because of its polarity, it can do all those cool properties we talked about. But some things are non-polar, and non-polar things don't mix with polar things because they aren't able to do those hydrogen bonds and mix and all that stuff. They can't do it, so they don't like to. Phospholipids are kind of like a national treasure because you have a polar region and a non-polar region, and they're bonded together, which I don't know how that gets like manufactured in a cell. I just know it is, and it's like a national treasure because normally it doesn't do that. But when it does do that, what's it called when you can have a polar region and a non-polar region? This is just kind of trivia. When you have a polar region and a non-polar region. Amphipathic, very good, Callie. Okay, that's awesome. So it's amphipathic, which is good because then you can put these phospholipids in these sheets where all the non-polar tails are together because that makes them happy, and all the polar heads are on the outside. So you get this bilayer. Now, is that bilayer permeable? That is, can everything just go through it? Semi-permeable, not everything. Very good. Okay, so the bilayer is semi-permeable. What kind of stuff can go through it? Small non-polar stuff. You guys are rocking it. Woo! I love how quickly we're answering today. Okay, so small non-polar stuff. Very good. Big polar things are not going to go through that membrane, at least not straight through the membrane. They're going to need another mechanism. Now, you may notice I have a couple GIFs on here, one of which looks like an alien wasteland. It is not an alien wasteland. That is just what your cells look like, which is kind of terrifying. But your membrane is very wiggly. It's like, woo. Y'all ever seen that TikTok where it kind of freaks me out, but it like makes your face look like a wiggly line or something like that? People use it to like spin out the door and stuff. Like that's what your membrane is like, and it's supposed to be that way because the membrane is flexible. It's fluid. And that's a good thing because remember, you're made of cells. If you see my face down in the corner, hello, it's me. But like my body, my arms, my cheeks, my nose, if I put pressure on them, they don't break. They don't automatically like burst into a puddle of dead cells because my membranes are flexible, so they can take a little bit of pressure. Whereas if we were rigid, every single time I tried to move, my cells would explode. This is a little bit off topic, but this helps some of my students remember. Have you ever heard of how, and this sounds a little ridiculous, but sometimes they will build buildings where they are able to wobble a little? That sounds really silly, but it's true. Okay, so buildings wobble, there's a reason for that. If you build a building too rigidly and it has no give, if something like an earthquake happens or something, very good Shreya, or a tornado, and it's too rigid, the building will just fall over because it has no flexibility. But if you let the building wobble a little, it may get hit by the tornado and rock and roll for a second, but then it may have less damage. That's the idea behind it. I'm not an engineer. I'm not an architect. I don't know the science behind that. It's just something I've heard. Your cells are the same way. They're flexible, so you can put pressure on them and it won't hurt them. Someone here said, I've been wondering for a long while, are we touching our cell membranes when we touch our face? I mean, yeah, I know like you have little hairs, like peach fuzz on your face. I have washed my hands, by the way. That's why I'm touching my face and I'm in my house. Like you have little hairs and peach fuzz and things, but I mean, your skin is just a bunch of epidermal cells. So yeah, that's just a bunch of cells. Everything living is made of cells, remember? So yeah, you're just touching some cell membranes. Your teacher may have done this with you, but if you ever look at the underside of a leaf, you can actually kind of see, if you put it under a microscope, like the itty bitty cells outlined on it. It's really, really cool. Okay, back to this membrane function, because we're already at 20 minutes and I've got several more slides. So we know it's a fluid mosaic. That fluid mosaic is a very key word here. So in addition to having phospholipids, what else is embedded in that membrane? Proteins. What for? Is it so you can get gains? Why do you need proteins? Why do you need cholesterol? What are they for? Transfer, transport. Very good, very good. Transportation. What's another reason you can have proteins in your membrane besides transport? Signal transduction. Ooh, identification. I love the multiple syllables. Yes. Okay. Homeostasis. Great. Ooh, big words. Cell-to-cell recognition. Yes. Get those big words. Okay. So this is not endorsing big words. Like you can use short words as well. I just like to hype them up because it's hard to remember all the big words in an AP science class sometimes. So yes, proteins in the cell membrane are going to be used for transportation, so they can move stuff through the cell, or they can be used for signals to like send out a little radar like, wee, I'm here. Or they can be for identification, so when something hits it, it can be like, hello, this is me. Or they can recognize cells. They can help maintain homeostasis by letting stuff transfer through. And it's really, really small. I'm trying to move my head so you can see it, but I don't. Oh, no. Come back. I went away for a minute. I'm sorry. But it's not letting me move my face. I'm sorry. So next to my face, right over there, you'll see all these little protein examples. You don't need to memorize all of those little proteins, okay? But those are some of the different reasons you can have them. Someone just asked, what is cholesterol for? It holds the membrane together. Very good. It provides a lot of that flexibility. It's also an antifreeze. So cholesterol is a lipid, and it doesn't freeze very quickly. So it keeps your membranes from freezing up and literally freezing. Like there's not a better word that I can think of. So I often think of membranes as having the consistency of like a bubble, kind of like SpongeBob bubbles, you know, where he can like poke it and it doesn't pop and it like walks around like a little bubble man, I guess. Anyone have any questions on this part? We're doing good on time. Okay, I'm going to move on. Okay, so let's talk about the actual transportation part that's coming into these membranes. So like we said earlier, this membrane is semi-permeable. Only some stuff is going to get through it. You can see where I've got my little pointer thing. Small hydrophobic. What's another word for hydrophobic? Nonpolar. Very good. So small hydrophobic or small nonpolar stuff is going to be able to get through this bilayer, just floating through on its own. So you can kind of see here on this bottom one, like a bunch of stuff is hitting the membrane and it's wiggly, like we talked about. This is a pretty accurate representation, but most of it's not getting through. The only thing that is getting through is this little blue and white one. So what must be some properties of that blue and white one? How is that blue and white one able to float through? Is it polar and small or non-polar and small? Small and hydrophobic, small and nonpolar. Very good. Okay. If it's polar, sometimes small polar things will get through, but not very quickly and not very efficiently. So it's not really an effective means of doing it. Can't water go through? So can water go through, or does water have to use a specific protein? Aquaporins. Very good. It needs an aquaporin. An aquaporin is a very specific protein that lets water through. Yeah, water is Very, 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 very polar. I'm sure you can squeeze like one water molecule through, but not enough to like sustain you. So big molecules, they aren't going to fit through just because they're too big. They're going to need to use, what process do we use for big or bulk molecules? It's another big, long word. If you want to put, that's close. I'm thinking of a different one. I'm thinking of like really big molecules. Engulfing, you're close. Endocytosis, very good. Endocytosis and exocytosis. So I don't have these up on the slide because frankly, I ran out of room because I hoarded all these images. But endocytosis and exocytosis are going to be for real big molecules, but they don't really play by the rules. They have their own little deal, which is great, good for them. When we're talking about membrane transport here, though, we're going to be talking about active transport and passive transport. So these top two up here, these are going to be passive transport. They just like slide on through like, wee, we're doing it, yay. Whereas the stuff on the bottom, it's not able to go through. They're not able to just simply diffuse through. They may be passive transport, but they may need that protein to get them through. So ions, why aren't ions going to be able to go through the membrane? They're charged. What's another word for that charge? They're polar. Very good. They're not nonpolar. Yes. So ions, they're not going to be able to fit through. You may have heard of something called the sodium-potassium pump. That's down here on the bottom. Sodium-potassium, the reason it needs a whole active transport pump is because those are charged ions. Okay. Getting a little ahead of myself. We'll come back to that. So keep in mind, you've got to know the concentration gradient. Does the concentration gradient go from high to low or low to high? High to low. Very good. Okay. And over time, it eventually will reach what point? Equilibrium. Very good. Okay. See, y'all are killing it. Okay. So eventually it's going to hit equilibrium. Passive transport is when it's just going to go down the concentration gradient. Does it require any energy? No. Very good. Active transport, how is it different from passive transport? Needs energy, needs ATP. What about, why does it need ATP? Why does it need the ATP though? Why can't it just like sneak in? Like, hee, sneaky, sneaky. It goes against the concentration gradient. Very good, Shreya. Very good, Derek. Okay, you're moving things the wrong way. Very good, Sierra. Yes. It goes against the concentration gradient. It's going from low to high. I always tell my students to think of it this way. Right now, there are 20 of you in a room with 20 desks. What if I took away 10 desks and asked all 20 of you to sit in 10 desks? I would be forcing you to get closer and closer together. And that would take a lot more energy from me, as opposed to if there was like a massive room with 100 seats, y'all would just naturally diffuse wherever you want to sit. Okay? What do we call passive transport when it involves a protein? Facilitated diffusion. Very good. Okay. What do we call diffusion when it involves water, the movement of water? Osmosis. Yay. Oh, y'all are so fast. This is great. Okay. We're 30 minutes in, so I'm going to go ahead and go on to the next slide. So we still have a full like 20 minutes to do the FRQs. Does anyone have any questions though before I move on? Nope. 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 Okay, I'm going to go on then. Nope. Ta-da! Okay. This is everyone's favorite one when we have to talk about tonicity. They're just like, why? And I'm like, because. But for real though, if you go to like nursing school, you do have to know this, or any form of medicine, because bad things happen if you don't match their like fluid levels. So that's why Gatorade is so important. It has electrolytes and Pedialyte and what's the other one? Powerade. So we're going to talk a little bit about electrolytes here in this tonicity review. Okay. This should be familiar because, again, this is a review. You often, when you talk about tonicity, you will see this diagram of like blood cells either shrinking or normal, like little donuts, or exploding. You'll often see these beakers that have this semi-permeable membrane. What else do we know of that is semi-permeable? The plasma membrane, the lipid bilayer, very good. The cell membrane, okay? And then we see how the water concentration changes, okay? So for example, if you look at this one on the left, it's got the same amount of water in each one, but this one has way more molecule. So do they have equal concentrations in here? No. But can the molecule fit through the membrane? No. So as a result, if only water can move through, water is going to move wherever there is more concentration to try to make it balanced. Very good. It moves wherever there's more solute. Very good, Derek. And so as a result, we now have an area that is less concentrated, but the area where the water left is now more concentrated, which is all good and fine until you end up with shriveled up cells, these hypertonic ones. So I'm pretty much just going to do the vocab here because, again, this is a review. I'm running out of time. But I also think it's critical that you understand this because they love to use this vocab. This is the kind of question that they love to make you apply stuff on. So let's talk about that. So if you've got a blood cell, which one is considered the healthy blood cell? Isotonic, the middle, very good. Sophia, you had a question. How can this flow of water relate to osmolarity? So osmosis is the diffusion of water across a membrane. Osmolarity, I get my vocab mixed up on this one, to be honest, when it mixes with chemistry, because osmolarity is going to be more talking about the number of solute particles per liter. That's more of a chemistry term. We don't get too much into, like, you're never going to be asked to calculate osmolarity. I would just stick with osmosis in this case. Does that help, Sophia? Okay, I think you might have just been mixing up a couple of AP classes, but it's okay. So isotonic is going to be our healthy one. This is what it's supposed to be like. This is when the environment surrounding the cell has an equal solute concentration to the solute inside. Are we going to go through water potentials? Maybe. You'll see. Foreshadowing. Hypertonic. Why is this cell all shriveled up? Why is it all shriveled up? Water, yeah. Because solvent exited it. Water moved out of the cell. Why did the water leave the cell? It's gone into the higher solute concentration. Where is the higher solute concentration? Outside the cell. There's more solute outside the cell to make the solute go inside the cell. Whoa, can the solute go inside the cell? Be very, very careful with that. They will tell you specifically on the question if the solute can cross the membrane, or they will say a large polar molecule, and you'll be like, that can't cross the membrane, because I know this, because I remember my membrane transport rules. So be very careful. When we're talking about osmosis, we're only talking about the water. Don't talk about the solute unless it tells you to. Only talk about the water. That is the safest way to make sure you don't cross yourself and lose a point. So yes, water moves outside to match that outside concentration. This is a hypertonic solution. It makes the cells shrivel up. But on the other hand, hypotonic, this is what happens if someone gets an IV of straight water with no electrolytes in it. As a result, you suddenly have way more concentration inside the cells. And so all that water flows into the cells, and the cells do what? They burst, they explode, they lyse, they implode. It's not good. So we would prefer our cells not explode because, you know, we like our cells. So anyone have any questions about this tonicity vocab? It's a little daunting. So please ask questions if you have them. Okay, I'm going to go ahead and go on. Dun, dun, dun. Let's talk about some labs. You may ask why there's a character from Lord of the Rings and a bunch of chickens. It's a little weird, but there's a reason, I promise. Okay, I'm gonna make my face big so you can see my expressions. With plant cells, it's a little different, right, because of the cell wall. Yes, so Japesh, that's a very good question. I should have put that on there. Good question. Plant cells, let me go back. Plant cells are going to be different. Now, they have a cell wall, so you still can have the cell membrane shrivel up from the cell wall, which is when a plant like wilts. But a cell typically won't explode if it gets too much water, usually. For example, like cacti. So cacti are super puffy because they absorb all of the water every time it rains. And as a result, that keeps them from dying when there's a drought. And their cell wall keeps them from exploding. So yes, Japesh, that was a very good question. Okay, gonna go back to the applicable labs now. You may be asking, why is there a weird egg factory and that Lord of the Rings guy? I don't get it. The reason that they are there is because the eggs, I want you to think of, your teachers may have had you do this experiment at some point, where you took a bunch of eggs and you put them in vinegar and that dissolved the shell of the eggs. And so then they had you like put them in different solutions. So you would see the eggs or the cells like grow and shrink as a result of that tonicity. So that's a lab that you've done before. Some of you may have done before that they love to talk about on the AP test. I've got an example coming up in a minute. For some reason, they also love to talk about potatoes. There's a lab where you like cut up potatoes and you basically do the same thing. The potatoes will like shrink and shrivel or swell up based on whatever solution you put them in. I think we did that in my class this year. If not, we did it last year. I can't remember. We did potatoes. Okay, thank you, Kenny. Thank you for keeping that together for me. Okay, so we're going to talk about these labs. And the reason I'm bringing them up is because a lot of teachers do them. And they popped up on the AP test, and I wouldn't be surprised if they popped up again, or if the skills you used on these questions popped up in the future. Foreshadowing. Okay, so let's go ahead and talk about that. So the first one we're going to look at here, it's a little bit light, so you may have to go up to your screen like this and just kind of look at it, and that's okay. Guys, I can't see you. It's okay if you look ridiculous. Y'all can see me and see how crazy I look, so just focus on me instead of yourself. Oh, there's nothing in my teeth. That'd be awkward. Okay, so a student conducted an experiment to determine the molar concentration of solutes in chicken eggs. Eggs were soaked in vinegar to remove the cells, so this is that lab. The eggs were then placed in sucrose solutions of different concentrations. The data is shown in the table below. So we see these sucrose concentrations. Initially, this is the mass of each egg, the final mass, the concentration of sucrose they were put in, the change in mass, and the percent change in mass. Okay, so pretty close to what kind of lab you would have done in class, okay? So the first thing it asks you to do here is construct a graph. I love when FRQs tell my students to construct graphs, because graphs are a lot of bang for your buck in terms of points, because you usually get 3, if not 4 points just for making a graph. And graphs are application, but they're not as strenuous application. It's a very good break for people who need a break from all the writing, where you can just like plot it and be like, boom, boom, axis done. So plus it's worth a lot of points. So I always tell my students, go straight to the graph. Most of the time, all you have to do is plot what's happening in the table. You just have to make sure you plot the right part. Then there will be a question when they ask you to explain it and predict something and da-da-da. But those are usually only worth a point or two as compared to drawing the graph, which is like half the points. So always draw the graph. So in this one, it asks you to construct a labeled graph on the axes provided to best illustrate water movement in and out of the cell. Okay. Then you're going to explain why there is a difference in percent of mass change in the eggs and the different sucrose solutions using the data, the molar concentrations of the chicken egg cytoplasm, and justify your answer. Another thing I tell my kids to do, and this would work if you had this like on paper in front of you so you could like circle and highlight and stuff. I always tell them circle the bolded words because that's where you figure out how many points things are worth. So for example, I already told you constructing a graph is usually going to be about 3 points. How many points do you think an explain type question is? 2. Okay. What about a determine? 1. What about justify? 2. Okay. So this is probably going to end up being an 8-point question. The bulk of those points are going to be in the graph. So I always say do that first. Okay. It's very easy to get partial credit for a graph, even if you're not quite sure what you should be putting on it. So I always tell my students, do the graph first. So this is a really good example of a type of lab question where you've done the lab. And this is the knowledge that we've talked about. So you need to talk about how water is moving. That's the tonicity. You need to talk about the percent of mass change. So how much water is moving in and out. Okay. You need to justify your answer. You may notice there's a little blank here. This is a really old question. And so it asked a question about like how this would work in the anatomy of a plant. To my knowledge, the AP curriculum does not cover plant anatomy anymore. So I did not ask you that question. So if you've seen this before and you're like, she left stuff out, that's why. Okay, I'm not going to go over the answers yet. I want your brains churning. So I'm going to skip a couple slides here. Okay, now we're going to take a look at the potato one. So we have another table. We have some more stuff. So now we're looking at water potential in potato cells. So they determined it by taking the initial mass of 6 potatoes. Then they took the cores of the potatoes and put them in sucrose solutions of various molarities or various concentrations. The masses of the cores were measured after 24 hours. Percent changes in mass were calculated. The results are shown below. Okay. So if you have a 0 molar sucrose or your control, it had an 18% change in mass, 0.2 molar had a 5% change in mass, you can read. So on here, it doesn't have it in bold, but what are the words that are going to tell you how many points you're going to get? Graph. What's the other one? Find. Very good. So how many points do y'all think this question is worth? How much do you think graphing is worth? 3. How much do you think finding is worth? 1. Okay. Find, I think, is going to be worth 1. We'll look at the key in a minute and see, but I'm pretty sure it's 1. Okay. So this is an example of that potato experiment. Of these experiments, the egg or the potato, if you've done that experiment before in class, would you please put in the comments egg or potato or neither? Just so I can like kind of get an idea of who's done this. Egg and potato, neither, potatoes, eggs. Okay. Potato. Okay, so a bunch of you have done at least one of these before. Okay, good. That makes me feel a little bit better. Self-agent, so no labs. I'm sorry. That's not fun. So these are 2 labs that I would say about half of you, maybe 2/3 of you are done or are familiar with. And so that makes it slightly more relatable. I want to look at one more lab before we start going over those answers. And so this is one, this is a really old one. I think this question's from like 2000 or something. I don't know. That's why the font is so different. So, but this question is also probably the most straightforward, do the science, it's the most mad scientist-y, I guess you could say, because you're not playing with eggs or potatoes. They give you like, and this is the one where they give you the dialysis bagging, and you open it and you put different solutions in. So in this experiment, they've got 4 solutions of sodium chloride. They put them in dialysis tubing. Oh, we did that too. Who else has done this lab? The dialysis tubing lab. Kenny, Smitha, Japesh. So this is back at the beginning of the year when teachers have all this energy and we're like, labs, labs, labs. So that's why this unit has so many labs. So, and this goes back to, I'm going to go to it and show y'all real quick. It's this one. If you've ever seen this question before where you've got a bag that's got water in it versus a bag that's got water and glucose or something like that. So it was on the test. Yes, Kenny, it was on your test. I think I put it on your test once or twice now that I think about it. Pretty sure it was on the semester exam. But anyway, and so this one, you've got to, again, do a graph. Are y'all seeing something consistent here? All of the graphs? Oh, thank you. That makes me so happy. Okay, so you've got to do all the graphs. How many points are those graphs going to be? Oh, Kenny, thank you. Okay, so the graph's going to be 3 points. You're going to draw and label 3 additional lines. How many points do you think that one's going to be? Let me make this a little easier. How many points for the draw? Okay, how many points for the label? Honestly, I'm just guessing. That's totally okay. 1 for each, okay? Now for C, our word here is an explain and include in your discussion water potential. Ooh. Explain, it's 2, very good. Include a discussion of water potential, probably 2. This is a long question, okay? I don't think they would have one quite this long on the new AP exam because that would take up like half of the test, but you never know. So let's go ahead and talk about these 3. We've got about 15 minutes left. So now you've seen the 3 types of questions that I like to put on there. I want you to notice when I see a lot of words, I get scared. Ditto. The only reason I look semi-okay with this is because I've been giving this question on a test for like 5 years. So guys, if you ever think your teacher seems overly confident and they're like, oh, they think this is so easy, but it's not, keep in mind your teacher has usually been teaching this for several years, or they had these questions when they were in school and they've been seeing them since they were 16. So it's intimidating. I was very intimidated by it in high school, but the more you familiarize yourself with it, the better. The thing that I learned to do when I was I didn't learn it till college. I wish I'd learned it in high school. So I couldn't do this obviously on tests. There wasn't enough time. But when I was practicing, because I would find old questions and try to work them out, I would rewrite the question into words I understood. And I would have to literally go sentence by sentence. Like I would cover words until I got to those sentences because it was just so overwhelming. Like I said, you don't really have time to do that on an actual test, but doing it to practice, it helped me just kind of get more comfortable with breaking things down in my head. Eventually, I didn't have to write it down. I could just read it. Yeah, I know. And that's why the idea of it being an open book isn't really helpful, because it's like, there's so many words. How are you going to even know which word to look up in the book? So, okay. Let's go ahead and look at these answers, though. Okay, so for the first one, on the eggs, chicken eggs, to be clear, they're not like platypus eggs. So remember, you had to make a graph, explain why there's a difference, determine the molar concentration, justify the answer. Okay, so for part A, to get that graph, this graph was actually 4 points. Usually, I see graphs that are 3 points. I would imagine that most graphs you do on this year's AP test would be 3 points because it's going to be such a short test, but they can be 4 points. And you get points for putting titles on your axes. You have to put the units. If you don't put the units, they can't give you credit. Like I had a question that I graded as an AP reader last year, and there were so many kids who didn't put the unit, and I couldn't give them a point. If you graph it wrong, do you still get the points? Because the rubric doesn't have the correct graph. You can get partial credit. So for example, let's say like the line is supposed to go up, but you draw a line that goes down. You won't get the actual line graph correctly plotted. That's this 4th bullet point here. But if you say labeled your axes properly and you did do the right data, you just plotted it wrong, you would still get those points. So like I said, graphs are great because you can get partial credit even if you mess up or if you don't have time to finish it, which is nice. Okay. The other thing I want you to notice is, so obviously you need to pick the right data. So the molar concentration versus the percent change in mass, you have to put the axis and data scaled. Okay. Standard error bars. So I'll be honest, like I haven't seen a question with standard error bars in a while. And the ones that I did, they're all very old. So I don't know if they're still making you do that. I doubt they would make you do that this year just for the sake of time. But obviously, if you know how to do it, I would remember how to do it. And if you don't know how to do it, it may not hurt to find a Khan Academy video. Okay, part B, we only have 10 minutes left, I'm sorry. So part B on the explain. So remember here, we had to explain why there's a difference in the percent of mass change. So you could get 2 points, but you had to talk about completely one of these to get one of the points. So it says the egg gains mass if the solute concentration is lower outside the egg, hypotonic, causing water to move into the egg. As an AP reader, what I will tell you, they are very, very, very picky about wording. They usually, in their head, have a certain word they want you to use. My guess would be that that is hypotonic. They want you to tell them hypotonic or hypertonic, but you can't just say hypotonic. Okay? You have to say, oh, this one is gaining mass, therefore it's hypotonic. This one is losing mass, it must be hypertonic. That means it's shriveling up. You've got to pick the word, but then you've got to explain, yes, exactly. Sorry, that you have to explain the why. Give the word, give the why. That's why, so like all through this chat, I'll ask you guys questions or whatever, and then I'll ask you more questions, and then I'll be like, yeah, but why? Yeah, but why? And I'm pretty sure most people think I'm like so mean and picky, but the reason is exactly, or else it's vague. Penny, I've taught you so well. Otherwise it's vague. AP readers don't like vague. So all of my kids, it drives them nuts. I have a stamp that says vague. And if they don't put enough information, like if someone just put hypotonic on my test, I would stamp vague on it. And that means they didn't get the point or the credit. Because you don't get partial credit for this. Now that is a cool stamp, vague. Yeah, it's in all caps. It's like vague. The only thing is I wish it had like red ink, but I don't really want to incur more trauma on people. But there's a reason why we do that, especially if you're trained to grade an AP test. If it's vague, they won't give you credit. And it's infuriating when you're the one grading it. And it's like, I know you know it. You just didn't give enough information. And I get that these tests are so quick. It's very hard to do everything all at once, but that's why you've got to study and have everything memorized so that you don't even have to think about it. It just pops in your head. That's my spiel. Okay. Let's look at part B real quick. So part B, the correct molar concentration based on the graph is 0.32 molar, 0.3 mole to 0.5 mole. If you put anywhere in between 0.3 mole to 0.5 mole, that would have been acceptable. And so here's their explanation for it, because again, you have to explain it. This explanation was a point. Whenever y is 0, that is whenever there was no sucrose, so it was like the control. There was no change in mass, no net water movement. That meant it was isotonic. So the solute concentration inside the egg and the outside the egg are the same, okay? If we have time at the end, I will come back and try to look at that math with you guys, but we've only got like a few minutes left and I really want to get the rest of this with you. So does anyone have, there should be a video on Fiveable on the math for this unit. So I would go and look at that. But I've only got 6 minutes left and I got 2 more questions. Any other questions about this question? I am not doing the cram sessions. I'm sorry. I really wanted to, but at the time my schedule was so packed, I could not. Now, however, my schedule is much lighter, so I kind of wish I could still do them, but oh well. The person who is doing them is amazing though, so you definitely should still look at doing them. All right, let's talk about potatoes. Potatoes. Yeah, Miss Coffee is great, so definitely go to her cram sessions. Like, they're awesome. Okay, So water potential in potato cells was determined in the following manner, blah, blah, blah. They weighed them, they exposed them to salt and sugar, da, 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 they changed. Okay, got that. So this one, basically the exact same thing. You're graphing it, you're finding the molar concentration or molarity. Da, 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 da. Okay, so you get 3 points maximum for this question. It's a very quick question. This is probably the kind of question I would expect on this year's AP exam because it's very, very, very quick. But like I said, we don't know that yet, but this is kind of like what my brain is telling me. So you're getting 2 points for the graph. You only get 2 points for the graph in this circumstance. So the graph isn't quite as rewarding in this circumstance. Still valuable, but all your points for like having the axes, the labels, the scale, and the units are just a single point. Having the proper data points, that's just a single point. Only 2 points for the graph. To determine the molar concentration of the potato cells, you can have anywhere in a range of 0.25 to 0.4. So I want you to notice that on both of these, they give you a pretty wide range. Like on this one, it went from 0.3 to 0.5. On this one, anywhere from 0.25 to 0.4. So if you're a little bit worried, you're like, oh, I need to round this. When it comes to calculating something from a graph, it's okay. I feel like they're going to make the questions really hard if it's open broke, like cure cancer or something. I don't know. I honestly don't know if they're going to be super hard or not. I don't think any of us will know until we see them. And I won't even get to see them until like July, which is infuriating. Okay. Last question. Okay. So this is the one where we have the dialysis tubing, and over time, and notice, be very careful here. Notice that this one, we're only looking at the 5% solution. Okay. And we're looking at it outside of the bag. Originally, was there any sodium chloride outside of the bag? Yes or no? No, very good. Okay, so over time, in this case, sodium chloride is moving outside of the bag. So in this case, we're looking at diffusion. We're not just looking at osmosis. This is not just water. Okay, so so wait, hang on. Yes, and it says here, like I said earlier, it will tell you if it's permeable to a certain thing like water, sodium, chloride, Okay, had to double check that. My eyes are playing tricks on me because my contacts are still in. Sorry about that pause. Okay. Like I said, this is an older question, okay? And as a result, I thought the calculated molar concentration came from putting the potato in the solution where its mass changes the least. How do I identify concentration in the graph? Talia, let me get through this question and I'll try to look at it super, super quick, okay? Because I do want to show y'all. Okay. So when we're looking at this particular question, this is another one where you're getting 3 points for a graph. So because it asks you to graph the data. So you get 3 points for the dependent variable and the independent variable. You get, and remember guys, always make sure you put your independent variable on the x-axis, your dependent variable on the y. That's the thing, it's like dry mix. Always do that right. They get real picky about that. Correct axes, scaling, units, and then plot it right. So that's 3 points. Now this is a long question because part B, where you had to make a prediction and label new lines, is up to 4 points. Wow. Now, here's the thing, though. They are kind of giving partial credit on this one. And this is why I gave this one so much in my test, because it did let me give partial credit. You get 1 point just for predicting where the lines are going to go. So in that case, you had to take the what do you think is going to happen with the 1% and 10% and 0%? And so they gave a point just for drawing those lines and predicting it. But if you explained each one not vaguely, they would give you another 3 points. But if your explanation was general, that's another word for vague, you only got 1 point. I have never seen another example of AP giving like credit like this. And like I said, this question's from like 2000. So don't anticipate that happening a lot. And then finally on part C, it asks you to apply it. So like if you've got plants that are growing near seawater, what's going to happen to those plants? Why is it going to result in crop loss? And that is because you are going to have, oh, no, come back. That was dramatic and then it didn't happen. Okay, so in that case, you're going to have to talk about water potential, how it's going to be hypertonic versus hypotonic, which one. You're going to be looking at how that water is going to move in or out of the plant cells as a result of that salt water. So if you want to get the full 4 points, you really need to go fully in depth into how that would impact like photosynthesis, transpiration, how it's going to cause the tonicity to change. You got to go in depth. Again, this is an old question, and the AP test this year is not going to have that much time to do this much for this many points. So I wouldn't anticipate this sort of question on the test. I would anticipate shorter questions that pull things like this, but I wouldn't be like, oh, it's going to look exactly like this. But the reason I showed you these 3 examples is because I can see them taking bits and pieces from each one. Does that make sense? Yes. Okay. So let me pull this part up right here. Okay. I want y'all to notice something. When it tells you to determine the molar concentration, on here, when it tells you to get the molar concentration, it says based on the graph. Based on the graph. It's not asking you to calculate it. I cannot change the focus on the crowdcast. I'm sorry. On my screen, it looks, oh, hang on. Yes, I can. Ah, is that better? I forgot my face was so big and that was ruining it. Okay, so let me show you all that one more time. Notice it says correct molar concentration based on the graph. Read from the graph. You are not expected to do math on this. Don't waste your calculator time on this. What they're wanting you to do, I'm going to make this big again, is that trick you may have seen before, where like you've got a graph and it's got a line. So like, let's say I've got a graph like this. It's wanting you to do that thing where you can like go bloop and find it. So that's what they're asking you to do. You're not supposed to necessarily do a math equation for this. So obviously I don't have time to go and plot every single graph on here with you guys. How do we know what type of graph to make for this? I believe this one's a line graph or any FRQ in general. So I'm trying to think, unless they specify, they are pretty much always line graphs. Because I do remember explicitly I gave one test where it said, make a bar graph. And a lot of my kids made line graphs. And I was like, it says right here to make bar graphs. It will pretty explicitly tell you if it wants a bar graph. Otherwise, it's almost always going to be a scatter plot. Does that help? Yeah. Most of the questions that I've seen, at least most of the recent questions, they tell you what kind of graph to make, or it's exceedingly obvious, like it's a change over time type thing. Okay. It is 4 minutes past, and I am so sorry. I have another thing I have to do. So I have to hop off. But guys, y'all have been awesome. The replay for this should be posted tomorrow morning. I would say today, but my computer's really slow at downloading it. So I'm going to try to make it start downloading it tonight. But I hope this helped. That way y'all can study in that. Be relying on your labs. I really think those are going to inform FRQs this year. But you guys did great. The participation was amazing. And yeah, I hope you all have a great night. I hope you're all doing very, very well. And see y'all soon. Bye. Bye, Kenny. Bye, everybody else.